Animal cell. Dangal greetings, Ma'am Charisse. To start, let me introduce myself first. I am Kaizen Claire Bartido, and together with my group members Chris Jewel Edesa, Celine Antonia Luna, Elijah Shane Netura, and lastly Carl Lawrence Rijo, we are Group 2 from STEM H4. And to tell you more about our cell model, here is Miss Jewel Edesa to start.
Animal cells. Animal cells are eukaryotic cells. They have membrane-bounded organelles such as mitochondria, and animal cells' mitochondria have their own genetic material, known as mPDNA. They are involved in energy synthesis by the means of citric acid cycle. Nucleus. The nucleus is where all of the cell's activity, including metabolism, growth, and reproduction, take place at. Lysosome. It's a single membrane structure containing various digestive enzymes. And here's Miss Elijah Nitura to explain further about animal cell organelles.
Golgi apparatus, it is involved in lipid transport, glycolization, and the packaging and secretion of biomolecules like proteins. Endoplasmic reticulum, an interconnected network of flattened sacs or tubules. Rough ER is the ER that is studded with ribosomes, and those without are called smooth ER. Peroxisomes. It breaks down organic molecules by the process of oxidation. Animal cells is a multicellular cells, and during cell division, animal cells divide by forming a cleavage furrow to separate daughter cells. This is in contrast to the cell plate that the plant cell forms during cell division.
Reason why we choose this cell type is because we choose this because of its distinctive characteristic that despite the fact that an animal cell is multicellular, it still lacks cell walls. The fact that it has fewer organelles than a plant cell makes it the easiest for us to do as well.
Materials and methods. Here are the materials that are already at home. Foam paper, brush, glue, tape, scissors, paper, toothpick, and pen. And here are the materials we purchased. Illustration boards, styro, clay, paint, brushes, and plastic cover.
Methods. Step 1. To create the cytoplasm, first cut a circle out of the illustration board and press clay onto it. Step 2. After creating cytoplasm, we created the cell membrane, which is also made of clay, and then we created each organelle individually. Next. Foam paper is used to create the microfilaments, microtubules, intermediate filaments, and flagella. They are simply cut into short and long straight lines. Both smooth ER and rough ER are constructed of the same color foam paper and have the same structural elements. The only difference between the two is that rough ER features dark spots, which are ribosomes. Nucleus, ribosomes, Golgi apparatus, mitochondria, centrioles, and lysosomes, peroxisomes are made of clay with different shapes and colors. Step 3. We write the names of the organelles on paper, then cut them out and place them on toothpicks. Step 4. The organelles have been arranged and labeled individually.
Realizations while doing your project. Due to the current situation, they weren't all able to come except for my one groupmate who lives near my place. My realizations while doing our project is that my groupmate is talkative and also creative. Even though we are far apart, all of my group members cooperate and willing to assist to finish this project.
As a person who doesn't have a sense of art but loves science, I realized a lot while conducting this project. I realized that cells have their own beauty. We use different colors to show the different part of the cell, and that makes it more beautiful. I also learned the different parts and functions of animal cell. Doing this activity made me realize that it's possible to combine art and science. Using our creativity and knowledge of science, we are able to create 3D model of animal cell. Having new faces as my group members, I also realized that it's really important to communicate and to have teamwork with your group mates to have the best result.
Doing this performance task and studying more about cells taught us many lessons and information that made us interested more in it. We learned the inner structure of a cell through this performance task and also saw the details of it. I also realized that my groupmates are kind and are willing to help each other.
I've discovered that my group members are simple to get along with after we created our animal cell model. I also fully comprehend the components of an animal cell. And here is our 3D model of animal cell. And here are the reference. That's all. Thank you.